Texas Pseudomycin treatment. Trying to do splicing. Primary urine. And what? Almost. Yeah.
Well, good evening, everyone. My name is Ed Campbell. I'm a graduate senior here in the Department of Biological Sciences here at CSU Stanislaus. And for the past 2 years, I've been awarded an NIH-funded 8-week summer internship through the Genome Institute at Washington University School of Medicine in St. Louis, Missouri. This past summer, I worked in the lab of Timothy A. Robert. And in this lab, we investigate 2 disorders of the blood. One is acute myeloid leukemia, otherwise known as AML, and the other lesser known one is myelodysplastic syndrome, otherwise known as MDS. So MDS is a clonal stem cell disorder characterized by ineffective hematopoiesis. Hematopoiesis, if you guys didn't know, is illustrated in this picture on the right. It is a formation of all the cellular components of the blood. All the cells of blood originate from hematopoietic stem cells, shown on the left right here. These stem cells differentiate and proliferate into multiple cell lineages that then mature into the cells of blood. In MDS, this hematopoietic process fails to happen, and these cells fail to mature into red blood cells, white blood cells, and lymphocytes. This results in patients presenting with anemia, which is a low blood count of the red blood cells. They're more prone to bleeding because they don't form the platelets needed to form clots. And they're more prone to infection because they don't have the lymphocytes needed to initiate a immune response. Often, MDS progresses to secondary AML, which is often a poor prognosis for patients because this particular type of cancer is chemotherapy resistant and is often a death sentence to anyone who receives it. So as a result, treatments for this vary. There is only one effective treatment, and that's a bone marrow transplant. But even this isn't 100% effective, and patients often progress to secondary AML anyway. So our lab took it upon themselves to try to investigate the genetic causes for this MDS. So we did this by using whole genome sequencing of MDS patients. And we compared the cancer genome to the somatic genome. And the way you use this is you take a sample of the bone marrow from this patient, which has the tumor cells in it, and you compare this genome to a sample that's taken from, let's say, the skin, a normal somatic genome of a person. And you compare the two and you look for any mutations that are present in the cancer genome that are not there in the regular genome. And we did this and we found a novel point mutation in this patient in the gene that codes for U2AF1. We then screened 150 MDS patients within the hospital and found that 8.7% of them contained the exact same mutation within the exact same mutation. So this mutation is located on gene for U2AF1. U2AF1 is an auxiliary splicing protein involved in splicing. If you guys know what splicing is, it's the process that undergoes in all eukaryotic cells, the processing of pre-mRNA to excise out introns, to leave just the exons that then leave the nucleus and then get translated to proteins. And this is the essential process that happens in every single one of your cells. U2AF1 specific job is to find the AG splicing site, the AG3' terminal end of the intron. So it comes in and finds exactly where this intron end attaches and then mediates the action of all the other proteins within the splicing. This mutation is found on position 34, located a little bit above my finger. It's black here, but it's arrow right here. In here we have all the genomes of humans on top and various other species on the bottom. And you can see that in all these other species, this same position 34 is located by located by S, which stands for a serine, a particular type of amino acid. And we see that in every single species we have a serine. So this indicates that this particular location is highly conserved throughout evolutionary history. And usually when something's highly conserved throughout evolutionary history, it means it's very important. Because if you change it, usually it's lethal for the organism and they don't make any more kids. So we see that this is highly conserved, and this is exactly where we find our point mutation. It results in a change from the serine to a phenylalanine. So, of course, the C2AF1 is involved in the splicing complex shown here. It is one of many proteins that are involved. We have E2F1 located right here at the 3' end of the intron. We have a whole bunch of other proteins that are also involved. And since our discovery of this mutation, other labs around the world have also discovered mutations within the same splicing complex, albeit in different locations, all linked to MDS. So this kind of shows us that, yes, we think our mutation is linked to MDS, and all the other genes are also linked to MDS also. In all the cases we only see one mutation arising in those patients. So if we have the E2F1 mutation arising, we don't see an SF3B1 mutation, for example. We only have one mutation. And we think that the accumulation of one of these mutations within the cancer genome results in the tumorigenesis and regression to MDS and AML in these patients. So we hypothesized that since we only see one mutation within all these patients, that a cell can only handle one mutation, that this one alteration gives rise to tumorigenesis, but any other alteration on top of that will be lethal to the cell. And so we hypothesized that this could be an effective treatment for patients with this particular type of mutation by inducing a second change in these cells on top of mutation that they already harbor. And so we hypothesized, or kind of thought about doing this using something called a splicing modulator. And these particular splicing modulators are called pseudomycins. So pseudomycins are synthetic analogs of naturally derived products that are obtained from Pseudomonas. They bind to SF3B1 and inhibit the function of that particular protein. And if you guys remember the last one, SF3B1 is also a member of the spliceosome. So this has the same function as E2F1, or the same role. So when pseudomycin binds to SF3B1, it is inhibited and kind of functions the same way that we think mutation functions. And so we think that we hit this genome twice, with the spliceosome twice, that this will be selectively inhibit just the cancer cells and leave the normal, unmutated regular cells of your body unaffected. And of course, that's our hypothesis that we want to investigate. So our hypothesis is that this S30F E2F1 mutation alters pre-mRNA splicing, and this alteration due to the mutation can be modulated by pseudomycin. So the way we went about investigating this is we used 3 vector constructs. Right in the middle, we have an MSCV virus, and the business end of this is a GFP gene and a U2F1, either wild type or the mutant form. If you guys know what GFP is, it stands for green fluorescent protein. So when you put these in the cell and the cells express it, it actually expresses the green fluorescent protein. If you shine a laser on it, it actually grows green, and so you can actually tell which one actually took up the vector construct. So we have these two with the U2F1 wild type and U2F1 with the mutation, and we also have the vector for control. So we took mice and we dissected out the femurs and the tibias and extracted the bone marrow from these cells. And we did CKIT positive selection, just to select for the hematopoietic stem cells within this bone marrow. And we cultured them up. And we did 2 retroviral transductions with these vector constructs we just described. So this introduces these genes into the cell. And by expression of GFP, we could tell which cells took up the DNA and are actually expressing the proteins that we want to express. So we did this twice, 24 hours apart. And then we sort for just the cells that are expressing this GFP. 24 hours after that, we transduce these, we culture these transduced cells with either 75 nanomoles of the pseudomycin, our splicing modulator, and we also do it with DMSO, or as a control, for 2 and 6 hours. And then after that, we isolate the RNA, prepare the cDNA from this. So we take the RNA from the cell and we change that RNA to cDNA, the coding DNA, which does not have any introns or anything like that in it, just the exons. And then we ran this DNA into with an RT-PCR assay. So one gene we looked at in this RT-PCR assay is FMR1. FMR1 has no biological relevance to MDS. It is just a gene that we chose because it undergoes alternate splicing, and we could actually measure alternate splicing that goes on. So I designed this primer assay. The first primer sits on exon 17, and and the second primer sits on exon 18. And when we run this PCR app, we get 3 possible transcripts. Transcript A on top results in normal splicing. So splicing occurs right at the junction of the exons and introns, and we get a 160 base pair product. And then we have 2 alternative splice products, B and C. This occurs when splicing undergoes at these cryptic splice sites within exon 18. So when splicing occurs at this cryptic splice site right here, we result in a 120 base pair transcript. And in the second cryptic splice site, we have a 85 base pair transcript. And so we wanted to find out what effect does just the mutation have on alternative splicing within these cells. And so these cells were not treated in any way with either DMSO or pseudomycin. These are completely untreated cells, but they have been transduced with either the vector, the wild type, or mutant E2F1. And we see the banding pattern in polyaggregate gel, where we have 160 base pair on top, 120 base pair on the bottom, or in the middle, and 85 base pair on the bottom. And for our purposes, we'll just look at the bottom 2, the alternative splice transcripts. And so when we look at the 120 base pair transcript, we see a similar usage between vector and wild type. This is expected because E2F1, the wild type version, should undergo the same splicing that normally happens, which we see compared to the control. When we introduce the E2F1 mutation, the use of this cryptic splice site increases significantly in these cells. This also occurs in the 85 base pair unit, where we have similar levels of usage in the vector and wild type, and an increase in usage after introduction of this mutant E2F1. And so we can conclude that E2F1 mutation does increase cryptic splice site use in FMR1. And so now we want to see how does pseudomycin affect this alternative splicing going on in these cells. And so we treated these cells for 6 hours with 75 nanomoles of pseudomycin, or indicated by the plus sign right here, or DMSO was controlled, indicated by the negative sign in each of these vector constructs. And just like before, we had this 160 base pair unit on top, and the 2 alternative splice on the bottom. So when we have the 120 base pair, if we just look at the DMSO control, the ones on the left here, we see, just like we did in the untreated cells, similar results, or similar levels of usage of the cryptic splice site in the vector and the wild type, and an increase in the mutant. After 6 hours of pseudomycin treatment, to the right on each of these graphs, we see a decrease in the usage of that cryptic splice site in each one of these vector constructs. In the 85 base pair units, we see the same thing. After 6-hour treatment of pseudomycin, we see a decrease in this cryptic splice site use. So this concludes that, yes, the mutation does affect alternative splicing, and this alteration can be modulated by 75 nanomoles of pseudomycin. And just to reiterate the point, we also looked at MDM2. This is also a gene that's biologically relevant. And we saw that exon skipping, another type of alternative splicing, is increased actually after 6 hours of pseudomycin treatment. And so, just like FMR1, we see that pseudomycin is actually modulating the alteration in alternative splicing to the mutation of E2A1. In addition to FMR1 and MDM2, we looked at these genes. So FMR1 was a tryptic splice site, undergo this type of alternative splicing. Genes that went undergo internal retention, MDM2, ReoK3, MDM1, etc. We also look at exon skipping genes like MDM2, fetch, and ENO1. So the summary, E2F1 mutation increases aberrantly spliced transcripts, and this can be modulated by pseudomycin. So this assay can detect alterations due to both. That was one of the goals of this project, was to actually develop an assay that can detect both these changes. Because we're using biologically relevant genes, so we want to use RNA sequencing in the future to actually find the genes that we think are causing MDS, and to put it through this assay to see how alternate splicing is undergoing and how it can be affected by pseudomycin treatment. And based on these results, we think that splicing modulators warrant further investigation as a novel therapy for patients hardware and the utility computation. Thank everyone there. Any questions?
So is it good or
Andrew, so is your hypothesis that the disease is caused by a new mutation, or is it passed from generation to generation?
We think it's caused by a mutation. We think it's a rise throughout life. It just pops up. Because this mutation is not found in any of the somatic cells, that's how it was discovered. We just sequenced the genome and compared it to your actual genome and we found that the difference. And usually these two diseases present later in life. We don't see it happening in children or anything like that. So this disease does come later in life. Yeah.
And then, so you have to focus in on looking at the effects on alternating splicing. Sort of pulling back a little bit, looking big picture, sort of clinical manifestations that we can present. What's the next thing you need to figure out to actually then be able to get from the point your research is at now to you've got this disease, take this pill?
A lot of things actually. So developing a treatment like that takes many decades, and this is some of the and so right now we want to actually identify what gene is actually causing it. So we don't know that yet. As of now, we don't know what gene is undergoing changes to actually get this. Because we know the Capstone mutation, but this mutation affects every gene that can be expressed. So we don't really know what gene that's actually that directly responsible. So we need to do that and find a way to stop that change. And then once we do that,
Step back 10 yards, sit down and wait for your brother to finish.
So I'm just trying to make a connection of what this accomplishes. It looked like you were studying a cell that's